Each year, Microsoft Research hosts hundreds of influential speakers from around the world, including leading scientists, renowned experts in technology, book authors, and leading academics, and makes videos of these lectures freely available.
I'll get started with my introduction while the last couple of people are coming in. My name is Katja Hoffmann. I'm a researcher here at the Machine Learning and Perception Group. And today I have the honorable task of introducing to you Antonio Cremonesi, who is a colleague in the same group. Antonio is a principal researcher. He's been with Microsoft Research for
a long time.
A long time. And today he will give us an overview of some of his latest research. When I asked Antonio why he's focusing on healthcare, he said that because machine learning for healthcare is the future. There are some very interesting developments there where we have experts on the one side, medical experts, who are very, very good at interpreting the data that they see and the information that they get from models. On the other side, we have machine learning algorithms that don't really have that expertise, that can very, very effectively extract patterns, but don't really know what to do with the patterns that they have extracted. So Antonio will give us new insights in how to make this combination work and generate very interesting insights from the combination of human expertise and machine learned models.
Thank you very much, Katja. Thank you for being here. Come on in, come on in. Problem is that lunch was too good. Yes, find a seat. Still coming in. That's good. All right. Thank you for being here. So you're stuck with me for about 50 minutes. Feel free to interrupt and ask questions. It's a very informal talk, very informal setting, so hopefully we will learn something from this. So I'm in the Machine Learning and Perception group working here in this building. And indeed, in the past 5 years or so, I've been applying some of what we know in the machine learning and computer vision space, applying it to the automatic analysis of medical images. And with medical images, I mean any medical image. So it could be X-rays, computer tomography, magnetic resonance, or even images of the patient being captured from the outside as well. So I'm assuming right now that we know very little about machine learning, and so I will have a sort of slow introduction, very pictorial, not much maths, to make sure that we're all on the same page and we understand the basic principles around machine learning. In particular, machine learning applied to the analysis of images. And I start with a very simple toy example. Imagine that we want to separate images of sharks from images of something else. It could be lizards. And so here we have collected some examples of sharks and lizards, and immediately as you look at those images, you say, oh, this task is really easy to do because I can just measure how blue the overall image is and be able to separate one from another. And that's absolutely true. For this toy example, I can represent each image in a very simple, very low-dimensional feature space like this. I can compute how much red I have in the image and how much blue I have in the image. And these, each of these squares represents one of these images. This is actually computed from these very images. And so you can see that separating these images becomes a very trivial task. Of course, this is a very simple toy example just for explanation, but computer vision is not so easy, and figuring out the content of an image, that it actually contains a lizard or a shark, is never so simple, especially when we move on to a large number of classes and more complex images as well. And so typically what happens is that you want to represent images in a much higher dimensional space. And as an example, this is again a three-dimensional space, and we measure 3 features rather than 1. So when the task becomes really, really complicated, it is impossible to design features by hand and hope to be able to capture an entire image or video even in a low parametric space like this one. So we have to resort to machine learning. And so what does that mean? It means that we measure some measurements out of the data that we have, in this case images, but we also have to have some labels. So this could be what is called the training set, and the labels here are represented by these colored circles, where blue indicates class 1, and this tan color represents class 2. And the whole purpose of machine learning, well, I have to say a very specific class of machine learning, which is in this case supervised machine learning, is to learn a generic function. Where is it? Here we go, this green curve that separates in some optimal sense the 2 classes. Now, of course, we want the separating function to work well, not just for the training examples that are these squares and labels, but also for any new image that I haven't yet seen. And so that's the problem of generalization. So this is pretty much what was done for Kinect. So when you go and buy Xbox and you buy Kinect and you start playing Kinect games, machine learning is what drives everything. In what way? Right, so the Kinect depth sensor produces these sort of images where each pixel is associated with a gray value, where the gray value indicates how far that 3D point is from the camera. And so you see different shades of gray from dark to bright, where dark means close to the camera and bright means far away from the camera. And what we've done is we collected a very large repository of depth images, where each of those images, for each pixel in this case, we had associated a label that told us whether that pixel belonged to the right arm or the left arm, the right hand or the left hand, the right side of the head and the left side of the head, and so on. We had 31 different classes for the different body parts. And so what we did is we trained a classifier that would associate a pixel in the depth image and its context as well. It's very important to look not just at the individual pixel, but what surrounds the pixel as well, and be able to predict a per pixel class. Earlier we were talking about a prediction per image. In this case, is per pixel, so that's a big difference. It's a lot more computation. And so all of this was done with some classification algorithm that was extremely efficient. Now, when I talk about classification, again, that is a subset of all supervised machine learning. There are many supervised machine learning algorithms. Classification is one subset. So how exactly was that done, and what type of classifier did we use? So this very woolly iconic representation of a classifier now becomes a decision tree. So probably most of you know what a decision tree is a data structure which comprises a root node and then some split nodes or internal nodes, and then some leaf nodes, also called terminal nodes. What does that have to do with classification? We have probably learned that this can be used to represent very efficiently data, which is of a hierarchical nature. How can I use this for classification? Well, imagine again that what I have is a picture. This is a photograph of 2 of my many kids, and we want to learn to figure out whether this picture represents a scene that was taken indoors or outdoors. So you can say, oh, I know how to do this. Is a really simple task. I'm very proficient at MATLAB or C++. I can write this in no time. And so I start writing down a code, and I think with my human brain, and I say, oh, the first thing I can do is I can check whether the top half of the image is blue. If it is blue, that's likely to be sky, therefore it is likely to be outdoors. And so effectively what I do is I can start constructing a list of nested if-then statements, right? So the first statement is the top part of this picture blue or not? If it is, then I travel here, and then I say, oh, wait a minute, I should perhaps also check if the bottom half of that picture is blue, because if it is also blue, then perhaps I'm just staring at a blue wall, you know, so maybe it is indoors. And so you start writing these, you know, if statements, one after the other, one after the other. Every time you test your algorithm, there are always cases where it doesn't work, and then you think, oh, okay, there is an exception, and you have another exception. And effectively, you end up with a very, very big tree of if-then statements, and that's your program, right? And unfortunately, it never works. Sorry to break the news, but computer vision is a lot harder than that, and every time you think of a rule, there is always an exception. It's a little bit like Latin, for the ones of you who studied Latin. Same thing. So how do you fix this problem? You fix it through machine learning. So what we want to do is we want to collect a lot of, you know, images which have been labeled by humans with what we call ground truth labels as being indoors or outdoors. And we stick with the decision tree type of architecture structure, but we want to be able to learn, you know, the questions to ask and where to ask them, and also the structure of the tree itself, how deep, if it is balanced or unbalanced, you know, where to ask questions, what type of questions, all of that has to be learned automatically. So you can see that machine learning, in some instances, is not very different from, you know, writing stuff yourself in a program, except that, you know, the parameters of this program can be all learned automatically. And this is particularly true for one side of machine learning, which is the discriminative type of models. Oh, that's the wrong keyboard. I can use this. Okay, so let me walk through an example of, again, another toy example. We'll have a lot of toy examples to try to make yourself, you know, feel familiar with, you know, at least this type of, you know, machine learning techniques. So imagine that we have a two-dimensional representation of, you know, data. So we have x1 and x2 are my two coordinates, my features, if you want to call them this way. And we have four classes, color-coded, you know, green, red, blue, and purple. Apologies if blue and purple are not very easy to distinguish. So what we can do is we can try to learn a decision tree that separates those classes in a way that can be used and produces generalization. So what we do is, you know, we start by sampling completely randomly a hyperplane, a line in a 2D case, and ask the question, okay, does this line separate, you know, the classes well, or does it not? Well, it goes through all the blue points. It doesn't do a particularly good job. So we can randomly sample a different line, and we can say, does this separate, you know, my data well or not? And we can keep, you know, doing this for, you know, until we get bored, pretty much, or depending on how much computational power we have. Eventually, we find a line, which in this case separates, you know, two classes from two other classes in a decent way without cutting through any of those clusters. And so we say, oh, okay, I like this line, I'm going to keep it. And so you associate this line in a high-dimensional space, it's a hyperplane, with the root node. So that is the first test. And we say, this works well, I'm reasonably happy with it, I, you know, fix it, I freeze it, I store it into the root node, and now I proceed. So what happens now is I send all this data to one child. If we assume this is a binary tree, we have only two children, and then all this other data to the other child, you know, left and right children. And so I proceed. And so now I test some new hyperplanes, and you see that these hyperplanes are only half planes. They stop, you know, at this boundary because we know that boundary already. I keep, you know, testing all these different hyperplanes, and I say, oh, okay, I found another one that works well. Now I look at the other one recursively, and eventually I like this one, and I stop. And they don't need to be absolutely perfect. They don't need to be absolutely pure. And even if there is a little bit of impurity left there, you know, we can deal with this because, you know, we will see soon that, you know, we can use multiple trees rather than, you know, one single tree. And every time we test the new hyperplanes, you know, when I said we can check whether it works well or not, you know, this is done computationally by measuring an entity which is called information gain, which is a function of the purity of each node, which can be computed as the entropy. So probably most of you are familiar with this. It's just a way, and not the only way, to measuring whether the training data that arrives inside a bucket is relatively pure or not. So this is not new. I mean, a lot of people have been doing this sort of work, and decision trees are very, very old techniques. The difference here is that in recent years, predating Kinect, people have been applying decision trees at scale with lots and lots and lots of data. So we moved from a world where these decision trees were designed by hand. For instance, in the medical space, decision trees are still now used to say, oh, okay, my patient has got headache. All right, let's try this test. If this is positive, then let's try this other test. And if it is negative, we will try different tests. And so it's, you have this concept of hierarchy and therefore conditional computation. You run certain tests only if the previous chain is positive and not if it is negative. But now, these have been applied, at scale on thousands and hundreds of thousands, if not millions of images, like in the case of Kinect. And also, we have been able, thanks to advances in computer power, we've been able to test different variants of decision trees. For instance, one important element of the decision tree is the split function itself. So earlier, I gave you an example where we were using this sort of split function, so oriented, hyperplanes. But actually, in Kinect, in order to get, the most out of the small computational power you have in your Xbox, we use something which is even cruder and simpler, which is axis-aligned, hyperplanes, which really corresponds to taking one feature at a time and comparing it to a set threshold. So this reduces the complexity of the model, the number of parameters that you have to search over. But it will have some drawbacks, and we will see in a minute what those drawbacks are. On the other end of the spectrum, we can think of associating each node with a more and more complex split function that could be, a curved, surface in this, very high-dimensional space. So these are just some of the many things that you might want to change in your decision tree model. Eventually, when you train a model of this type, this is what you end up with. You end up with a tree that looks a little bit like this. So you have a root node, and then split, lots of split nodes. The tree can become very large very quickly, which is a problem. In this case, we have 16 or 20 levels, something like that. And the tree is not necessarily balanced. So there will be some branches which grow a lot further than others. Just because your information gain tells you that the nodes are still very impure, there is still information to be gained by splitting the data further and further. Once you have trained this a tree like this, and then you freeze it. Okay, you put it on your hard disk, and now at runtime you test it. So the test time, what happens is that a new image that has never been seen before is pushed through the tree. And like I said before, the first test is applied. You read off the test that was stored at the root. It is applied, and then this input data gets sent to the right child in this case. And then here a new test is applied. It gets sent to the left child, and so on. And it navigates its way through this hierarchical structure until you reach a leaf. And at that leaf, what happens? At that leaf, during training, you would have pre-stored the statistics that you have observed, the empirical distributions that you have gathered from the training data itself. So in this case, it says that the training data that arrives at this leaf is mostly green, of the green class, and then there is a little bit of red class and a lot fewer of the blue class. And so during test time, you reach that leaf and you say, oh, okay, this is the probability associated to that test input data, say it's an image. And then, what happens is that you can have multiple trees. So that's the key idea of a forest. So a forest is nothing but a collection of trees. Now, all these decision trees have been trained, say, in parallel, on a parallel architecture if you want, and therefore they can be trained all, very efficiently. and with some level of randomness. So, while you train the trees, you make sure that, each of them individually produces some, good level of, prediction, but at the same time, they're all slightly different from one another. And the fact that they're slightly different from one another, helps you in achieving higher generalization, higher generalization for the data, test data that hasn't been observed beforehand. So how do you combine the outputs of these many trees? For instance, here's an example. Imagine that we have 4 trees, and imagine that right now we're in a regression setting rather than a classification. Regression means that your prediction is a continuous value. We call that value y. And V is my high-dimensional input feature vector. So each tree effectively computes the conditional probability, of Y with respect to the input vector. And imagine, for ease of argument, that at each leaf we model this conditional probability as a Gaussian. So it's a Bell function that looks a little bit like this. So for the same input data, you push it through all 4 of the trees. They will reach 4 different leaf nodes, and at each of these leaf nodes you will read out a different Gaussian distribution. And let's imagine that they are, these are the 4 different Gaussian distributions. So they're all normalized and they're color coded as corresponding to the 4 different trees. And what you notice is that some of them are more peaked, around a certain mean value. Some others are more widespread. So this indicates the fact that some trees for that data are much more confident about the prediction than others. Now, typically, the way you merge together different individual tree predictions and combine them together to get a single forest prediction is very simply by just averaging out these distributions. And if you do that, you end up with this black curve where what you see is that the highest confidence tree has got the most weight, the most mass in the final forest prediction, while the ones which are very non-confident, very uncertain, will have much less influence on the final prediction, which is precisely what we want. But of course, simple arithmetic averaging is not the only thing we can do. We can do geometric averaging, multiplication between the different distribution, and this would lead to very different behaviors for the forest itself. Now, all I'm trying to say is that we have done a lot of work in analyzing how things change, how the behavior of the random forest change if you change different parts, different submodels within the tree model. For instance, what happens if you increase the depth? So here is another slightly more complex toy example. Again, we have a 2D space and we have these 4 spiral arms representing 4 different classes, and we need to learn to separate those classes. Now, just on the training data, all these models work perfectly well, right? They do 100% score. But the behavior outside of the training area, like near the corners of this square, are all very, very different. So you see that if we use the sort of split functions that we used in the Kinect application, where you have axis-aligned features, you start to get very weird, blocky sort of artifacts. If you use oriented lines, oriented hyperplanes, you have a level of generalization that, at least visually, people will feel a little bit more comfortable with. And then if you use conic sections, you have probably an even better behavior, especially if you look at this corner here compared to this. You see that here you get more gray, and the gray color indicates higher uncertainty, which is precisely what you want to get as you move farther away from the training data. So what I'm trying to say here is that, as many of you would know, it's not important to just achieve 100% accuracy on the training data. It is also important to look at the accuracy of my confidence, right? So how confident am I, when I move away from the training data? and what is the accuracy of my predicted confidence. And that is a very tricky thing to measure. It's very, very difficult. But at least here we have some visualizations that allow us to make sense of that. There is code available. You don't need to buy my book, but you can download the free version of it, so don't worry about that. And the code is also freely available for research purposes. And both the book and the code are designed with first-year PhD students in mind, and so hopefully it's all very accessible. It gives you a nice, gentle introduction to machine learning with applications to computer vision and medical image analysis. Now, let me talk about some variants of decision trees and decision forests. One variant which is particularly interesting to us is what we call jungles. Sorry for the lack of imagination. So what happens with decision trees is that, although they are very useful in practice, they are still limited. In particular, one way in which they are limited is, this combinatorial explosion. So as the tree grows, I use more and more levels of depth, which is, having big trees is useful in real life applications like, computer vision, like, semantic segmentation of images, for instance. As it grows, the number of nodes you need to store and you need to run computations for, grows, with the power of 2. So it grows very, very quickly. But also, an even bigger problem is the fact that, quite quickly, we run out of, training data. So we have a training set which, has got a limited size. It gets pushed into the root node, and then, in average, it gets split into half and half. Half of it travels to the left sub-branch, another half. And then, you subdivide it into half, 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 half again. So you have quarters and so on. Very quickly, you just run out of training data. So you cannot grow these trees, to a depth that you would find it really, ideal. So what do? Well, a simple idea would be to try and merge some of the nodes together. So the outputs of this node and this node get merged into a single node rather than having two separate nodes. And now the question is, okay, that's an interesting idea because it allows us to use the same amount of training data more efficiently, but how do I merge things? And of course, if you have multiple of these new structures, which are not trees anymore, but these are directed acyclical graphs, they're called DAGs in short, an ensemble of those is what we call a jungle. So they help, both because, now we can constrain the memory use, we can constrain the maximum number of nodes we have in the DAG and in the jungle. And hopefully, because we reduce the number of overall parameters controlling this classifier, that might have also positive effects to what is called generalization. I'm not going to go too much into the details of that, but happy to discuss it with you. So let me explain a little bit, again, with another computer vision-based toy example, how this might work. So imagine that we have images of sort of pastoral scenes, like in Grantchester, and we have animals of two different types, cow and sheep. And looking at patches of these images, we want to figure out whether those patches belong to the grass class or the cow grass or the sheep class. And of course, we want to be able to capture appearance variations within those classes as well. Like for instance, the grass in this case appears very bright, very saturated dark green, and in this case is very bright, almost brownish, yellowish. So let's imagine that we represent these patches in our toy two-dimensional space, where we measure the X, the brightness, and here some form of chromaticity. Then these patches correspond to these points. And let's imagine that we have collected many such training examples. So you might be able to see some very faint ellipses. Here is one for all the cow patches, here is one for all the sheep patches. And then the grass, because it's got very wide variation in appearance, we have some training examples here, nothing in here, and some in here. So if we train a tree, to separate these classes, we might start with this, sort of, split function at the root, and then we can proceed with the left child here, the right child here, and then we split off, these two children again, here and here and here. And what we will end up with is with this region having, again, the data that arrives in each bucket, is reduced very, very quickly. In this case, we have only one, cow patch arriving in this bucket, so this entire bucket, gets assigned the label of cow, and this one sheep. And clearly, visually, at least in this toy example, it doesn't make too much sense because, these cow and sheep live in between, training examples of grass. So this is not great. If instead, we use a DAG representation, we might start in exactly the same way as we did with the tree. This is exactly what we saw before. But now we say, oh, look in here. So this bucket here, contains mostly, grass example, and there is also, sorry, this is the wrong way around. There is also one triangle, which is a sheep. And this one also has got mostly grass example, and there is some, impurious other stuff happening. And so because of this semantic similarity, statistical similarity between these two buckets, I can decide to actually merge them together. And so this gives rise to a DAG because I'm merging nodes together. Because I'm merging now them together, now everything in between here, everything in this entire bucket gets labeled as grass. And therefore you can do better generalization within this sub-manifold if you want to think about it this way. So this is the final DAG, which does a more correct generalization and captures the illumination invariance, and this is sort of what it would look like in practice. So we have got this merging happening here. All right, this is all very toyish instructional sort of examples. Now I would like to spend a few minutes talking about applications. I'll probably go through these a little bit more quickly because there is a lot of detail here, but I just want to capture the gist of how these things can be used in practice. So one first application we worked on in the medical space is that of anatomy localization. So quite often in an emergency, in an A&E department of a hospital, people can come in, they're rushed in because they have been victim of some sort of accident, a crash. And so the first thing that happens is, the doctor doesn't know exactly what's going on, so they stick the patient inside a CT scanner, a computed tomography scanner, which produces this sort of images. Mostly, a big chunk of your body is imaged in this way. So these are called full body images, although they're not entirely full body. And the first thing they need to do is they need to search through these images to very quickly figure out whether you've got broken bones, whether something has moved around in ways that are not entirely healthy. And doing this with traditional tools is very time consuming, and time is very critical at that stage. What we propose here is a way to automatically analyze this image and just just navigate through the images by saying, oh, I want to look at the kidneys, I want to look at the spine, I want to look at the heart. Imagine having a menu where you can just click, click, click the different organs. In order to do so, the computer needs to understand what the organs are, what they look like, and where they are. And so we put together a system that, given a CT scan like this, it's actually in 3D, can automatically detect and zoom into the relevant organs. And we can do this very, very quickly, very effectively for roughly 50 or so anatomical structures. So it's very efficient. It takes only 2 seconds on a standard desktop computer to do so. The problem is very hard because we all look different, not just in the outside, but also in the inside. We have this preconception that, oh, this is easy because the kidney is a kidney. How different can it be? Well, some of us have got very tiny ones. Some of us have got very big ones. Guess what? And so, and the same is for even with being completely healthy, right? Of course, then there are lots of anomalies. Like in this case, there are massive cysts. In this case, there are other cysts somewhere else. The images can be captured with contrast agent or non-contrast agent. They might use very different scanners, very different resolutions, depending on how much time they have to capture the images, and so on. So we need to be robust with all those changes. So although in the first instance you might think that this is a much easier problem than classifying photographs into cows and sheep and penguin, it is actually still a very, very hard problem. And we use machine learning to do this. In particular, we use a form of decision trees, which are regression trees. I mentioned those earlier, where what we want to do is, given an anchor point, a key point which is easy to distinguish in the body, we want to be able to predict, the continuous variables which, from that anchor give us the position of, say, the bounding box for the left kidney or the right kidney, and so on. So all of this is done, using a probabilistic, version of decision trees, which at the leaves, encodes, these Gaussian distributions for the displacements of the 6 sides of each bounding box from each reference point. And the reference points themselves, the anchor or landmarks, whichever way you want to call them, they're also learned as part of this, end-to-end procedure. So this is what we end up with. We end up with our own 3D visualization of a CT scan, a full body scan. And these green areas, which have been highlighted here, represent the automatically learned anchor regions, which the system in this case learns to use because they produce the highest confidence estimates for the position of the kidneys. So when you see this spinning around again, perhaps I can make it spin around again, you see that, you see that these green regions correspond to the top of the pelvis and the bottom of the lung, which make a lot of sense because those are transition areas between, say, bony structure and muscle, or air and muscle, which are clearly very distinctive visually, and also they're very close to the kidney, and therefore, intuitively you think, oh, it makes a lot of sense to use these as anchor points to figure out where the kidney is. But all of this was learned completely automatically. So another application that has been quite successful in this work is something that, by the way, this is work that we've been doing with Adam Brooks Hospital, which is here in Cambridge, and it is one of the biggest research hospitals in Europe. So they've got masses of data, and they're very keen to collaborate with us at the research level. So we've been very lucky to be working with some doctors, radio oncologists in particular in this case, who not only they understand medicine inside out, but in their free time they play with MATLAB. So it doesn't happen very often. So in this case, we're talking about brain images. These are magnetic resonance images of the brain, captured with 6 different modalities. Each of them shows some different aspects of the tissue. In this case, even the untrained eye can immediately see that there is something really unusual going on in the brain. Unfortunately, that is a very nasty tumor. It's called glioblastoma, and it is incurable. And unfortunately, the unfortunate people who get it, they don't have much life left. There is very little they can do. One of the reasons why the prognosis is so bad is because doctors don't have very good ways of measuring whether a certain therapy is working or not. So although doctors are particularly good at looking at these images and say, oh, this is definitely a glioblastoma, they cannot say with accuracy how big it is. They cannot say whether the therapy that we have started 2 weeks ago is actually working or not. If it is working, by how much? How much is the tumor shrinking? Is it 1% or is it 10%? They do not have good quantitative tools. And so computer vision and machine learning can help in that sense, because, for instance, if we can segment out the tumor, and not just the tumor as a whole, but also segment out the different compartments, the different tissue types. In this case, in green, we have highlighted the necrotic core. Those are brain cells which are just dead. There is nothing you can do about them. Then there is the red area, which is called the active rim, the actively proliferating area. So those are the most active tumoral cells which draw in a lot of blood, a lot of oxygen, and proliferate very quickly. And then there is the edema, which is the surrounding area, mostly composed of healthy cells of the brain, but they are swollen. They draw in a lot of water. But unfortunately, in this area, you might also have some tumor infiltration already. And what happens is that each of these regions ideally should be treated with a different therapy. Some of them respond better to radiotherapy, some others respond better to chemotherapy. But they don't know this. They cannot extract these sort of maps. But now here we have a tool which is very promising. So what we do is we collect a lot of patient data. So each column here is for a different patient. You can see the variability of the tumor. These are, of course, horizontal slices through the patient's brain. And we also had experts label pixel by pixel these tumors in 3D. So that's very expensive data to capture, but we were lucky enough to get it. And then we train our decision forest. Sorry for the cheesy animation. And once we have trained it, we store it on disk, and then we can apply our runtime. So we have a new patient coming in with a tumor of the same type, but in a different place in the brain, with different appearance, with different ratios of necrotic core versus active rim versus edema. We push it into the random forest, and a classification comes out with a segmentation map. So we can say, exactly where the edema is, the necrotic core, and the active rim. And so we've done this for a bunch of test patients that were never seen before by the training algorithm. And these are the sort of correspondences. Sorry, these are the sort of correspondences that we get. So here we have the ground truth manual segmentation, which is itself not perfect because it was done by humans, and even experts might have some differences. And these are the sort of automatically obtained maps. So it's quite encouraging to see this working, and it works very, very efficiently because it's all based on random forests. These are some visualizations that for these are most useful for the non-experts, people like most of us, I suppose, that they are not medical doctors, to see the 3D shape of these things. But they're also useful for surgery planning. So I've been working with some surgeons who, looking at these 3D images, can better plan, what to cut and what to leave. So perhaps I have time to talk about a final project, and then we'll have plenty of time for questions. So another big problem in medicine is the actual data acquisition. So hospitals in the Western world, especially, they spend a lot of money, buying very expensive, scanners. So we have magnetic resonance scanners, we have PET scanners, we have CT scanners. They're manufactured by a few a few big companies like Siemens, GE, Philips, and some others. And each of them costs, many, many millions. So you can see that, and also the running costs are very high. So each patient that gets scanned into one of those scanners, has to be chosen carefully because it costs a lot of money. But also, especially with magnetic resonance, the time required to scan a patient is quite long. So it goes from 10 minutes, if it's not a major organ that they need to scan and not many sequences, to many, many hours, depending on what you need to do. In clinical practice, they try to keep the time, for scanning within 1 hour. But that means that a patient who is going through some, painful time of their life will need to be, stuck and completely stationary within a magnetic resonance, which is a very, small confined space for 1 hour. Okay, elderly patients or very young patients cannot really do that. So anything that we can do to acquire those images very quickly, would be, absolutely welcome. But of course, you can degrade the image quality, and get the scanning, done a lot more quickly. But what if we want to maintain the resolution, the high resolution of the images? So we came up with an idea to super resolve images. So acquire images. So this is a very special type of magnetic resonance image of a brain. In this case, it's called diffusion MRI, which effectively for each small volume in your brain, it measures diffusivity of water. think of this as, what is the principal direction in which the water, can move within a small section of, my brain. And that corresponds to fiber tracks, especially, in the white matter, you have very anisotropic, sort of structure. They diffuse water only along the fibers, the connective tissue in the white matter. In the gray matter, instead, is more like, mud. There is no structure in there. Having a good idea, a good visualization of these structures, the fibers, is very, very important to figure out what is connected with what, and especially if there is something like a tumor, it's very important to figure out what that tumor is impinging on and whether it is impinging on areas to do with speech or with motor skills and so on. So here you see a low resolution, very quickly acquired scan of a patient brain, and here is the same thing in much higher resolution. So this color coding indicates different orientations. So we can try to learn this mapping from low resolution images to high resolution images. These are all done in 3D, using this simple formulation. So we have a low resolution grid, the blue dots, and from that we want to predict the red dots, which are 4 times the size. And we do this using, once again, regression trees, regression forests. And these are very difficult to see, but here are the results of our forest interpolator. If you want to think of it this way, this is the original high resolution data, and numerically and visually a little bit, you can see that here we retain some high frequency information, sharp edges, compared to other techniques, and also numerically we show that we do better. And this is yet another very weird and not used in clinical work technique called NODI. It's yet a different type of magnetic resonance modality. So yeah, this is the numbers, all good. I want to conclude here so that we can have some time for questions, if you have any, by saying that this work is not only super cool, but also extremely useful, and things are changing. The landscape is changing as we speak. There is all sorts of crazy stuff happening out there with social networks and machine learning and artificial intelligence. But I bet that one, if not only one, of the next big things will be a revolution in the medical sector. There is huge amount of data which is completely untapped. Machine learning and AI techniques applied to that data really promises to revolutionize the way in which healthcare is delivered, the way and reduce the costs, as well as benefiting the patients as well. So if you're looking for a job, this is a really good area to learn more about and put your skills in, and I'm sure you'll do well. So any questions? Yes.
I have a question. Thanks. I have a question. The example, the presentation is super. It's great. The examples you gave are all related to a snapshot in time where you have a certain image and you look at the image and then you come up with something. How do you deal with temporal information that is more of a temporal nature? So you're following a certain case and you're following the developments, whether they're becoming better or worse based on the type of treatment that they get.
Yeah, it's an excellent question. So, looking at temporary studies is really where we want to go. We haven't done that yet, mostly because of lack of data. Really, getting the ground truth data is extremely expensive. Even for these single temporal snapshots, getting them for following through the patient is even more difficult. But also there are lots of challenges. For instance, in the case of the brain tumor, those are all pre-operative images. So these are the images that get acquired, when they come up with a diagnosis before any treatment is done. After the treatment is done, if it is radiotherapy or if it is surgery, the image will look very, very different. In particular, in the case of surgery, if the tumor has been resected, then you will be left with sometimes a really big hole in the brain. And also around the hole, there will be some scar tissue, which is, naturally it's a healthy process of healing. But unfortunately, if you image the patient again, that scar tissue from magnetic resonance is impossible to distinguish it from tumor. So it just looks exactly like tumor. And so you don't know whether the tumor is growing back already or whether that is scar tissue. And that is one of the biggest problems right now with this type of tumor.
For non-surgical things, it should still be applicable, shouldn't it?
Probably it will be a lot easier than what I just described, but we haven't done so. And definitely a very interesting area to look at.
Thanks for your talk. Have you included expert data in your featuring? Using as prior information?
So the experts, the medical experts.
Yeah, the specialists, yes.
The specialists work with us all the time, right? So this work cannot happen without having medical expertise. And it is very important, if you want to embark in this sort of work, to really connect with local hospitals who have got the right level of expertise. And we're particularly lucky here because, again, Addenbrooke's is one of the best. But I'm also working with hospitals in France and in the US as well. The experts give us domain knowledge, which is clearly super important, but also help us get the ground truth data, the ground truth labels. We don't necessarily want them to give us features, like visual features, because nobody knows how to design good features. We want the machine learning algorithm to extract the features that are good for the task. And this is part of the revolution that is happening right now in machine learning and computer vision, where you have all these deep convolutional neural networks. That the one thing they're really, really, really good at is really figure out automatically what features work for the task. Some of this automatic extraction, at least selection of features, can be done also within decision forests in a slightly different way. But we try to not to design the features too much because we're always wrong. It's a bit like writing your own if-then statements. They're always wrong. Okay.
Thank you.
Hi, thank you. Could you please elaborate on why decision trees are working that good in this problem?
In the problem
of the segmentation or even the prediction of sizes of the brain, different brain segments?
Sure. There are many answers to this question. So one of them is that decision forests are particularly efficient, both at train time and at test time, right? So at train time, being efficient, it means that you can try many different experiments very quickly. And if you have some high level, what's called hyperparameters to tune, then you can do that very quickly until, the problem, works. At test time, it is important to have efficiency because, the doctors are always in a hurry and the patient even more so. So that's good. From an accuracy point of view, it has been observed repeatedly that having multiple trees, produces very good accuracy, even on previously unseen images, which is really important to stress. So that, although individual decision trees have this problem of overfitting, learning too much about the training data and too little about, generalizing, decision forests are better in that sense. Having said all this, decision forests are not necessarily the only thing that works in this case, right? So having seen what is happening with convolutional neural networks, I have little doubt, they will work very well in this, case as well. In fact, we did do some experimentation with convolutional neural networks applied to 3D multimodality magnetic resonance images, and indeed they do work very well. However, some of you might have tried, using convolutional neural networks already, and training them is very complicated, very time-consuming. Figuring out what is the right architecture of the network is something that is quite painful to do. So there are advantages and disadvantages. Both of them would work in this problem, but there are still differences. I think there was a question in the middle there before there. Sorry.
I was wondering, do you see basically the loop close in the sense that the results from machine learning being used for the medical doctors to train to see kind of tumors in the images?
Yes. So one way in which I could see these sort of tools be used is, imagine that the doctors looks at the MRI of a patient using their standard visualization tools, which normally come within their, with their pack station. When they buy the scanner, they also buy some visualization tool. But then you can augment the visualization tool to say, oh, look, if you click this button, you can see my estimate, of where the tumor is and its segmentation map. And they can look at them and then they can say, oh yeah, I can see why that looks good. Or if it is incorrect, as it might be, then they say, oh, this is incorrect, this is wrong. And effectively, by clicking on a button which says this is wrong, that data is sent back to some cloud-based repository where it is labeled as, oh, the algorithm, messed it up. And so the expert algorithmic people like myself could look at the data and say, oh, okay, it is not working here, why is it? And you try to reason, you try to improve the algorithm and feed it back. And with time, the algorithm will become better and better. So this is, this feedback loop could be extremely useful.
But kind of also for the medical doctors, so you may detect something that the doctor doesn't see at that moment.
So that's why, especially in oncology, they have weekly meetings where they discuss the same case, multiple doctors at once, which is clearly a very expensive thing to do because time is money. But multiple oncologists, they sit around the same table, they look at the same data, and they interpret it together because of that.
Unfortunately, we're out of time, so I would like to ask you to take the remaining questions offline. I would like to thank you all for contributing to the talk with your interesting questions, and to Antonio for a very interesting talk.
Thank you very much.
Each year, Microsoft Research hosts hundreds of influential speakers from around the world, including leading scientists, renowned experts in technology, book authors, and leading academics, and makes videos of these lectures freely available.